So our guest of honor for the 24th Annual International Tinnitus and Hyperacousis Conference. We'll have a concert tonight. is Jennifer Melcher, and we're quite honored to have Jennifer here. She is a very smart person and also a very thoughtful person. In addition to that, she is very committed to helping tinnitus patients and understanding and finding treatments and cures, and very sincere in her approach. So because of all these factors, I've been impressed with Jennifer for many years, and she is our guest of honor for this year. Please welcome Jennifer.
So can you hear me okay? Yeah, so thank you very much for, that was actually probably the nicest introduction I've ever had. Thank you very much. It's a pleasure to be here. I've been meaning to come to this meeting for years, and it's nice to finally be here. I'm not going to be talking to you about treatments, and I'm not going to tell you what drug is best. I'm a basic scientist. For years, I studied the auditory system, not really looking at tinnitus, and then I started doing fMRI, brain imaging, and there was a colleague where I was at the Mass Eye and Ear Infirmary, very interested in tinnitus, and he was really keen to use fMRI to look at what was going on in the brain of people with tinnitus, and so we paired up, and we got some initial results that were very interesting, and I'll be telling you about some of that later. But that sort of began a bit of an odyssey that has taken me deeper and deeper into tinnitus. But always tinnitus in people, in humans. Quite a bit of sort of the more basic work of trying to understand the mechanisms of tinnitus is done in animals. But I firmly believe that controlled studies looking in sort of a basic way at what's going on in tinnitus have to happen directly in the species, if you will, of primary interest, which is, of course, people. So what I'd like to do is tell you about work that we've done, my group, I'm certainly not working alone, on people like this. So I like this picture because it conveys a couple of things. One is it really does convey the kind of person that we study. These are people who are out and active on their way to work and not majorly depressed, not holed up someplace. They're very much the person on the street, and that's who we study. Sometimes it's a person on the street who's come to the clinic and then come to us, and sometimes it's people who just come to us directly. And I also like this picture because it conveys the fact that tinnitus is a hidden problem. So these people are going about their daily lives, and they've got inside their heads various things going on. Some are hearing a sound, and that's about it, hearing various sounds. Some are hearing a sound that's particularly aversive. Others are hearing a sound that's actually related to a depression or a mood that's declined. There's sleep that goes along with the tinnitus, percept, and sometimes concentration, and all these other things. So tinnitus is not just, of course, everybody here knows, hearing sound that isn't there. It's a condition that has a lot of pieces to it. And of course, also there's the hyperacousis that can go along with tinnitus. So what our work does is look at both ear and brain. Rich did a great job explaining, it's basically those two things working synergistically to give us this, or give people who have it, this perception, sound when there isn't actually sound. And as I said, we're working on people, some of whom are patients. And where we sit sort of on the classic bedside to bench spectrum is somewhere in the middle. So on the bedside end, I think of people like Claudia, who's out there on the front lines, like Rich, treating the patients, all right, in a variety of ways. We've got the trials that we just heard about, and that's not us either. And at the other end, we've got the animal models and the neural mechanisms. So what we do is, and at least what we've done so far, is focused on understanding the mechanisms as best we can directly in humans and trying to make our experiments just as controlled as if we were doing an animal experiment, okay? Which is not actually an easy business when you're working in the messiness of humans. So much of what we've done and what I'll talk about today focuses on the auditory pathway. Again, Rich did a great setup to explain that there's various ideas about how aberrant neural activity within the auditory pathway gives rise to this sound in the absence of external sound. And I'll be coming back to this schematic, this diagram over and over again, so let me just be sure everybody's on the same page. It's basically showing an optimal slice, if you will, through the brain and the ear. So the middle ear, cochlea, auditory nerve, cochlear nucleus, and other brainstem structures, thalamus and cortex. And the black, of course, are fibers. And I just want to point out these fibers, we often think of them as going up. And in fact, neuroanatomists like Brett Schofield, who have studied this intensively, and I'll come back to his work later, have said that there's good evidence that there's more coming down than there is going up. All right, so there's an opportunity for massive, deep, descending influences on the auditory system and the aberrant activity that causes tinnitus. So in our studies, what we've done is we've wanted to look directly in humans for elevations in spontaneous activity. Okay, this is the elevations in neural activity that Rich was describing form what might be the longest standing hypothesis, and you can correct me if I'm wrong, for the basis of tinnitus. Okay, so the auditory system, the the neurons are just sitting there firing away, and this gets amped up and heard as sound. That's one theory of tinnitus. Now, we haven't been able to look directly at the spontaneous activity of humans, and so we sort of make a supposition in trying to test this idea that there's elevations in activity. Some people will call it actually an increased central gain. We use sound-evoked activity with the idea that a lot of the mechanisms that might cause an elevation in responses to sound may also be at work in increasing spontaneous activity. And going back to where I started in all of this, it was the early days of functional magnetic resonance imaging, and we had just discovered a way to start imaging brainstem structures, in particular the inferior colliculus. And so we decided, let's look at the inferior colliculus in some people with tinnitus. All right? And tomorrow I'll go through the whole saga. It began in about 1998, sorted out the saga and all of the data in about 2009. Okay, so there were a lot went into basically finally determining that actually you put sound into the ear, you image the inferior colliculi, and what you see on average in a person, whoa, that does this, okay, in a person with tinnitus compared to without is more activity, more response to sound in these pea-sized structures in the person with tinnitus compared to without. All right, and so everything, and I'll go into this in more detail as far as threshold is matched between these people. They're both normal, clinically normal, audiometrically, and there's other ways of matching. This is not a hyperacusis effect. At one point we did think it was, but it's not. It is an elevation that's related to the tinnitus. So we've got this initial result, and the way we think about this currently is that it is an illustration of hyperactivity, an extra response to sound in the inferior colliculi, an example of increased central gain in people with tinnitus. So one thing I forgot to mention already is that, and I've used this as a scientist, I get cold calls fairly frequently from people with tinnitus. They want me to talk to them about their tinnitus. I have to tell them I'm not a doctor, but I can say to them, all right, I am a scientist and I can give you some possible explanation explanations for your tinnitus, and I can tell you about some scientific findings. And one thing I'll say is, yes, there really is something going on in your brain. You really do have a volume that's turned up. We have pictures that basically show that this is the case. All right, so some of what I'm telling you today, I offer to you as a means of talking to your patients about what is going on inside their head and to validate the fact that there really is something truly physiological that causes the tinnitus. All right, so starting from this point, what we've essentially done over the years is work down in the system and work up, okay? So some of our work, and that's what I'm going to actually talk about in this talk, has looked downward into the brainstem to understand what's going on in people with tinnitus, and some of it has gone upward into auditory cortex and beyond, and that's what I'll talk about in my talk tomorrow. So if we move downward, what I'm going to be focusing on here today is basically actually below the inferior colliculus, okay, so the bit that's outlined in purple there. So what's happened in the animal literature is that there's a huge amount of evidence that if you at least give an animal a hearing loss, you will see increases in neural activity in the brain. And that has been a very hot area of investigation in recent years. And so what I'm going to do, just to give you a backdrop, is just a very, what I'm going to say, almost superficial sampling of some of the animal data, okay, so you have a sense of that. And then, though, what I'd like to do is move on and talk about directly in people, what can we see? And the first thing is I'd like to give you an introduction to the people who we study. I gave you a little bit of an introduction, but I'd like to give you a little bit more of a feeling for who these people are in terms of their characteristics, audiometric and otherwise. And then finally, I'll get into what is going to be the bulk of what I'm going to present, which is tests for brainstem hyperactivity in humans, okay, using a sound stimulation. Okay, so Rich talked about having hypotheses that you can test, okay, and what I'm going to be presenting to you are some tests, albeit somewhat indirect because we're using sound, but of one of the hypotheses that Rich put forth. And I'm showing you off to the side this sort of a little more detail of what's going on in this purple outline. And it's just a diagram fleshing out a bit more this very simplistic view of the auditory system. So we have the cochlea, the auditory nerve. This block right here is the cochlear nucleus, which contains many different cell types. I'm showing you this in still a simplistic form. And these different cell types go off and project in all kinds of different ways, ultimately converging on the inferior colliculus and then obviously progressing upward, and then again receiving downward projections as well. The reason I'm showing you these details is that all of these different types of neurons with their different projections, they also have different neurochemistry. If tinnitus were to reside, if the extra activity were to reside in just one, it would be a target for the kinds of drugs that Claudia was just describing, okay? What I'm going to show you is actually the picture is very different. The extra activity or the increased gain is very widespread. It's through all of these very different neurochemical types of neurons, and that actually, I think, is food for thought in trying to think about how to go about treating tinnitus. And I'm going to try to come back to that some at the end of the talk. So I'm actually not going to be talking about brain imaging in this talk, because the best ways to tease apart what these different cell types are doing are using actually classic techniques that are used in the clinic all the time. We use them a little bit differently, but auditory brainstem responses, contralateral suppression of DPs, distortion product otoacoustic emissions, and then acoustic startle. And I'm going to present to you our findings using each of these techniques, as I said, in the main part of this talk. So first, just a little bit of background about animals. Basically, I want to show this slide because it's data of Jim Kaltenbach's, and it's sort of it sort of spawned really everything that's happening now in the animal literature on tinnitus. Back in the late 1990s, this figure says 2005, but it's taken from a review. In the late 1990s, what Jim found was that if he gave hamsters a hearing loss and he recorded from one of the major divisions of the cochlear nucleus, the dorsal cochlear nucleus, he just put an electrode on the surface and moved it across the surface. If he did those recordings before and after giving the animal the hearing loss, he saw what's shown here. So as he moved his electrode across the surface, which means he's basically recording from different characteristic frequency regions of the dorsal cochlear nucleus. This is what he saw in control animals that were not exposed. And 30 days after an acoustic trauma, this is what he saw in animals who were exposed. All right, so this is showing this extra activity, and this was the first time I think this was demonstrated in animals, extra activity, extra spontaneous activity in response to acoustic trauma. And this is something that evolves over time. It's something that involves plasticity of the dorsal cochlear nucleus, and you can see that because 2 days after trauma, the activity is actually reduced. All right, so since this time, particularly the dorsal cochlear nucleus has been an area of intensive study in animals. Things have moved outside the dorsal cochlear nucleus more recently. What Jim Kaltenbach did was just put a big electrode on the surface of the DCN, look at the spontaneous activity, but it was spontaneous activity in the same sense as an EEG is spontaneous. There was a big debate, went on for a while, about whether or not this extra activity was actually in individual neurons, and that's been ironed out. And this is showing increased activity in individual dorsal cochlear nucleus cells from a study from Susan Shore's group. And you can see that this is basically showing also that if you test an animal for tinnitus, and I'll show you in a minute how one way that people do that, you have this extra activity if they test positive for tinnitus, but not if they do not test positive for tinnitus. The other point, though, is that this extra activity, this increased spontaneous activity, isn't there for all the different types of neurons in the DCN. It's there for one specific type. Okay, so this gets into could it be that there's certain particular cell types in the brainstem that actually are responsible for or carry the elevated spontaneous activity that accounts for tinnitus. And this is just briefly showing, and I don't think we need to go through the details, a test for tinnitus has become quite popular. It basically involves looking for a suppression of an acoustic startle response. I'm showing it mainly because it comes back to the other reason for why we actually want to look directly at humans. At the end of the day, humans tell you whether or not they have tinnitus. Especially if you ask them the right way, you can be quite sure they have tinnitus. When we recruit our subjects, we make it clear that you're going to be paid regardless of whether or not you tell us you have tinnitus, okay? There's no incentive whatsoever to basically end up in a tinnitus group or a control group. So that's the other reason we look at people. All right, so just to finish out this animal work that I'm giving very short shrift, elevations in spontaneous activity have been seen in both of the divisions of the cochlear nucleus. This is in response to noise-induced cochlear damage. Inferior colliculi, the same thing, auditory cortex, even the amygdala. And in fact, if you want to get into the far-flung regions of the brain that really are involved in tinnitus, there's a recent finding demonstrating the importance of a particular part of the cerebellum in the development of hyperactivity in the inferior colliculus. Okay, so that is just to say that there's ample evidence for central gain, increased central gain in tinnitus that comes from animal work, with the caveats that the information is in animals. And so let's turn to humans. And so first let me just tell you about our people. This is a fairly typical tinnitus, happens to be a tinnitus subject. Most of what I'm going to describe to you today is our data that are in men, and that's probably something worth keeping in mind. We did that because as part of our attempt to control things, we wanted to make sure that major variables like threshold, sex, and age were all very well matched between any group of tinnitus subjects that we're looking at and any control subjects we're looking at. And we basically just didn't have the capacity to look at both sexes and do it well. So anyway, this is showing a very typical audiogram, and you can see that we are looking at the very people that Dr. Hansen said are very few and far between as far as tinnitus is concerned. And I guess I'm not really sure that I agree with that. I think it may be where you see them, whether it's in the ENT clinic or whether or not it's, in other environments. We see plenty of folks like this, and we call them clinically normal or near normal, but you can see that as you go above 8, these guys are rolling off in threshold, and we know this is very much real. If when we bring in our young 20-year-olds, we've got flat thresholds out to 16K. And of course, they're also, we're not talking about thresholds that are 0. We're talking about thresholds that are 10 and even 20, and we even let some of our folks drop all the way down to 30, okay, at about 8. So we're looking at people with normal to near normal hearing, and we're doing that for a particular reason. We sort of want to be looking at a group of people who are sort of on the cusp of becoming worse. We're looking at responses to sound. We want to see those responses to sound well. We don't want them to be complicated, either in terms of their audiogram or in terms of other factors like depression and anxiety, and that's why we're not looking at people who have major depression, for instance. Okay, so I already made these points. And then I actually don't have a slide describing the quality of the percept, so I replotted Stouffer and Tyler's data because the main thing to look at here are just the descriptions of the percept. Qualitatively, I would say that, we're especially in these top categories seeing the same kind of distribution, and certainly the same representation of more complex as well as sort of the classic ringing percept. And I should say that actually most of our people have bilateral tinnitus, and that's just to keep in mind, and that's different a bit from the statistics that Rich was showing, perhaps partly because we've been selective in terms of the audiogram and the way that, and we're sort of drawing subjects from a different source. Our people, most of them do not report hyperacusis when they walk in the door, but when you test them, on average, you'll find that their loudness discomfort levels and also their qualitative report of tolerance of sound is lower than a audiometrically matched control group. So these folks that are in this bar graph have audiograms that are matched at least up to 8K. I mean matched. I mean like the mean differs by maybe a dB at 500 hertz, the frequency of this stimulus. And this is a well-known phenomenon. Sylvia Hebert has also, in the similar kinds of people, published the same kind of effect. What else about our folks? Our folks they tend to be a little bit more depressed than their matched controls. And this is reminding me also to tell you that their age tends to be about mid-40s plus or minus 10 years. Okay, so it seems like that, early mid-40s, that's when tinnitus is all of a sudden become this issue or developed. And then they tend to be a little more anxious. But there are few that are really up here in terms of depression and anxiety scores, sort of in the major depression or anxiety range. So I'm showing you, and I just should say that these scores, oh, I took it out. Okay, never mind. So I'm showing you this, and I'm showing you the preceding slide, because what we have tried to do, at least in the sample of data I'm going to be showing you today, is be sure that what we're seeing is related to tinnitus, not related to depression, and not related to loudness discomfort level. All right, so these are, in a sense, for what's to come, confounding factors that have to be taken out of the equation to actually draw the conclusions that we're going to attempt to draw. All right, so now the meat of this. What I'd like to do now is just basically move through 3 studies that we did examining the function of these cell types using the methods that I mentioned before. So The overall goal is to test for brainstem hyperactivity and do this directly in people. And so we're going to do that using the auditory brainstem response, acoustic startle response, and contralateral suppression of otoacoustic emissions. And I'll point out to you as we go along that each of these is actually invoking or relies on a different subset of these neurons. All right? I should say that they rely solely, as far as we know, on neurons in the ventral division of the cochlear nucleus. So in some ways, what I'm showing you here is complementary to what has been explored most intensively in the animal literature, which is the functionality of the dorsal cochlear nucleus. All right, so what we're going to be doing is looking at tinnitus groups that are closely matched to control groups without tinnitus. As I mentioned, the groups are matched in sex and age and in hearing threshold. So this is an example of the match between tinnitus and control groups that we achieve when we are making comparisons of our various measures. And then finally, if we see a difference, say in wave 5 of the ABR between people with tinnitus and people without tinnitus, we play devil's advocate and we say, okay, could that difference be because of a difference in depression, anxiety, or sound tolerance? And so we check that in various ways. And I'm not going to go through the details of that for the most part, but that's been done after the fact. Okay, so auditory brainstem response. I think you all know very well, right, that it's produced by stimulus, and I'm going to be talking about click stimuli, presented to the ear. Auditory nerve responds and produces an initial peak, which is wave 1, and then there are subsequent peaks that are produced by neurons in the brainstem. And you record it. You can record it in various ways, but the most typical is between an electrode near the top of the head and then one, say, on the earlobe, ipsilateral to the stimulus. So we, as usual, had our 2 age and threshold match groups. And what we did is we said, all right, we're going to go in and we're going to quantify the amplitude of the ABR waveform. Okay, so we need to look at amplitude because we want something that indicates amount of activity, all right? And that means we also have a little bit tougher time. We have to do a lot of averaging to really make the baseline before the stimulus quite clean, okay? What we also do is we open up the filter. So we're looking at the ABR. It basically, the typical filter would have the peaks more down near this baseline. Things are riding on this lower slow wave, these lower frequency components, and that's actually important for seeing some of the differences that I'm going to describe. All right, so we pick off amplitudes as shown here. And the first thing is, let's just look at wave 1, which is from the auditory nerve. And it is worth noting that these folks, the tinnitus folks, are not quite identical to the controls, and it really fits actually with what Dr. Hansen was saying earlier, which is that, you know, essentially he was saying more hearing loss, more likely to have tinnitus, all right? And the picture you get from the nerve, from the ABR, says a similar thing. So down near threshold, all right, we've got 4 click levels here. We've got mean wave 1 amplitude for the tinnitus and the non-tinnitus controls. And down near threshold, these guys are looking pretty similar in amplitude. But as we go up in level, what we've got is an increasing disparity, and actually the tinnitus subjects are showing a little less activity, or a lot when you go to highest levels, compared to the matched controls. Okay, and that's despite the fact that these are matched groups. We had to do the matching separately for the different levels, but the story is the same. The audiograms are pretty much on top of each other. So what's going on here at these higher levels? Well, it comes down to the fact that when we look at an audiogram, when we look at threshold, we're not sampling, we're not using all of the auditory nerve, we're not looking at all of the activity of the auditory nerve. And when we're at threshold, and I should explain what this is showing is data from CAT. The threshold of different auditory nerve fibers plotted as a function of their characteristic frequency. All right, so down here, each point is an auditory nerve fiber. When we're at threshold, these auditory nerve fibers, represented by these triangles, are controlling the threshold. We have no information about all of these auditory nerve fibers up here, which don't respond at threshold. So what we think we're seeing in the ABR is essentially a loss or a lessened activity of these guys up here. So at the low levels, our ABR in tinnitus subjects is similar to the control subjects, but at the higher levels, they're different, and the tinnitus is less, perhaps because they're missing these higher threshold auditory nerve fibers. And this fits really well with the current sort of rage right now, which is hidden hearing loss. The idea that behind a normal audiogram, at least in animals, you can actually have, and in animals that even have completely normal complement of inner hair cells, they've been traumatized, they've been allowed to have their thresholds recovered, they have normal inner hair cell counts, they have normal thresholds, but their auditory nerve fibers are still missing, okay? So what we think is going on in some of these tinnitus subjects is that there is some hidden hearing loss. Okay, but what about sort of the meat of the matter, which is the hyperactivity and whether or not we have it? So this is showing a quantification of wave 3 and wave 5 at these same click levels. And you can see that there's an overall trend for the tinnitus folks to have higher amplitudes of both 3 and 5, and it reaches significance at a couple of levels. And if we do it again, new cohort of subjects, we see the same thing. This time, the more significant differences are in wave 3 rather than 5, but all the trends across all peaks basically stay. And I'm showing you this. Okay, so there's a couple of points I want to make. One is, so the results are basically consistent with the idea that we have a hyperactivity, an increased gain, in whatever neurons it is that are producing wave 3 and wave 5. It's not a resoundingly huge increase in gain, but there is more activity in response to sound in the people with tinnitus than in the people without. The other thing is that this result has been replicated, all right? We've seen it twice and we've gotten it twice. And in the field of tinnitus and in tinnitus research, this is really, really important, okay? Basically, I don't believe anything until I've seen it twice, and ideally, I believe it best when 2 different groups have seen the same thing and been equally careful, all right? It's very rare in the field for that to happen, okay? So anyone out there who wants to do an ABR study like this, I'd like to see this again tested for replication by somebody else as well. And it's important, especially if we're talking about small effects like this. The effects are small, but I think they're actually important, and that's for sort of, as I said at the beginning, reasons of thinking about treatment, okay? So let's go on to talk about, and all of this is happening despite the matched audiograms between the tinnitus and the control subjects. Okay, so what does this tell us about the activity of these cells in people with tinnitus? All right, so we already said we have a reduced wave 1, all right, reduced auditory nerve activity in the people with tinnitus compared to the controls, and it might reflect hidden hearing loss. But what we also have is elevated waves 3 and 5. And I'm not going to go into the details, but longer ago than I want to admit, I did a PhD thesis on the generators of the auditory brainstem response and made lesions in different parts of the cochlear nucleus and in the brainstem, and looked at, this is in cats, and looked at what the effects were on the ABR. And what we concluded was that the dominant contributors to the ABR are not just everything, it's particularly the spherical and globular bushy cells of the ventral cochlear nucleus, okay, and certainly not the dorsal cochlear nucleus. So when I look at a slightly elevated amplitude of wave 3 and wave 5 in people with tinnitus, I think, okay, turned up gain in some particular channels through the auditory brainstem. Okay, so that's the ABR story. Let's move on and look at an acoustic startle response. So The acoustic startle response circuitry has been worked out best in rodents that actually have a group of cells that sits in the auditory nerve, and humans don't have them. But what's closest to those cells in the rodent are the globular bushy cells. Okay, so those guys probably mediate the acoustic startle, which is through, ultimately leads to, in people, an eye blink. So put in a loud sound, you blink your eye. And so when we look at how much you blink your eye, the strength of the EMG activity, one way of thinking about it is we're looking at the amount of neural activity that's in this pathway. Okay, so here we go. We're looking at two groups closely matched, and this is showing the location of the EMG electrodes right under the eye. This is a classic procedure for measuring acoustic startle. We don't do it so much in the auditory field, but in the behavioral psychology field, it's out there all over the place. And so we just, and I shouldn't say we, really largely Inga Knudsen is the person responsible for these experiments. She decided to adopt basically the procedures that have been used just out there over and over again. What you see when you present a loud sound is electromyographic activity following the sound. This is showing 3 different trials, all at the same stimulus level. And again, sort of following the classic procedures, you can take the activity in a given response window, you rectify it, you smooth it, you rectify it, and then you can do a time averaging. You end up with a measure of the amount of activity that's produced by the startling stimulus. And you can take that and plot that as a function of the level of the stimulus, and that's what's shown here for a group of people without tinnitus and a group of people with tinnitus. And you can already see that there's this tendency for the red curves to be a little higher than these black curves. And again, we're looking and comparing people who are matched. They're matched in age, sex, and threshold. Now, for this particular, and I should have said this already, when we did our usual procedure for the ABR, we checked to make sure that there were no dependencies of wave 5 amplitude on LDL, on depression, anxiety, okay, and there were none, all right? So we're convinced that what we're seeing in terms of elevations in wave 3 and 5 are related to tinnitus as opposed to hyperacusis or somehow depression or whatever. And here, it was a little trickier because there's actually a very strong effect or strong relationship to loudness discomfort level. And this is actually, it relates very much to the animal work that's going on, and I won't get into that in great detail. I'm not going to go into that. But for purposes here, this dependence of the amplitude on loudness discomfort level had to be taken out for us to understand how much of this increased amplitude in the people with tinnitus is related to tinnitus rather than loudness discomfort level. So we regressed out loudness discomfort level. And what we found was if you look on average, still, with loudness discomfort level taken out, an elevation in the responses of the people with tinnitus as compared to those without. So we've got 2 measures now that are telling us that there's a volume that's turned up in the auditory brainstem. And this time, it's not spherical bushy cells and globular bushy cells. It's more likely the globular bushy cells, and then additional structures in the brainstem that I'll mention in a few minutes. Okay, so we've sort of got at least 1.5 channels that are showing an increased central gain. Now let's look at another measure that actually depends on very different cells from the first 2, okay? And this time, I actually need to flesh out the picture a little more and show you both sides of the auditory pathway. So we've got the cochlear nucleus on a side that is being stimulated by a noise, and then we have the cochlear nucleus on the opposite side. And what we're going to do is, and again, what Inga Knudsen did, all right, is measure distortion product otoacoustic emissions from this cochlea. So basically, what we're doing, as we see it in this experiment, okay, is looking at, what we're doing certainly in this experiment is looking at the effect of this noise on these distortion product otoacoustic emissions through the medial olivocochlear neurons. okay? All right, so what we're doing is, though, we're using the distortion product, the DPs, in this year as a readout. When we look at them with and without this noise, we're looking at that difference, basically, as a readout of the amount of activity in this pathway, okay? All right, so, and that's just the measure, those are the values we used if anyone's interested. All right, we matched everybody. We controlled for sound tolerance in a slightly different way in this study, which was we basically divvied people up according to their loudness, discomfort level, and sound tolerance on actually Rich's questionnaire. So we've got folks that have both tinnitus and non-tinnitus subjects who have very high sound tolerance, so they've got a high loudness discomfort level. They're very tolerant of sound on this questionnaire. And then we've got people who have a lower tolerance of sound. All right, and I'm showing you particular dividing lines that apply to the data that comes in the next slide. We move these dividing lines around, we divvy people up in different ways, and the results hold no matter what. All right, so, and we matched audiograms and the DPs themselves across the 4 groups that are defined by tinnitus and sound tolerance. Are pretty similar. Okay, so we got the DPs here, all well above the noise floor for the frequencies that I'll be showing data for. Okay, so what's shown here, plotted as a function of one of the frequency of the one of the 2 stimuli that is used to evoke the DP, is a change in magnitude. So this is the magnitude of the DP during the noise compared to the contralateral noise compared to the magnitude of the DP without the noise. Okay, so in one situation, we're actually stimulating that channel that crosses the brainstem, and in the other situation, we're not. And we're looking at the difference in magnitude. And the main thing to see here is that there's one group that's lying lower than the other 3 groups when you look across frequency, and it's the people who are very tolerant of sound. Okay, they've got high LDLs, and they don't have tinnitus. And these other groups are showing a little bit more suppression of the DPs in response to the contralateral noise. Okay, so in these top curves, that channel, those neurons that run from the noise-stimulated ear to the DP-recorded ear, those neurons are more active in these 3 groups, all right, with tinnitus and poor sound tolerance, tinnitus, regardless if they've got good sound tolerance, and then no tinnitus with a low sound tolerance. All right, the important thing, though, is we've got elevations in the 2 groups with tinnitus. And you can see that again here. We've just bundled up the data in half-octave bands. And again, this is the people who have no tinnitus and no problems with sound tolerance by any of the measures. And then most importantly, these red bars, which are our 2 groups with tinnitus. All right? So what we've got is more activity in this pathway that's mediating that contralateral suppression of DPs, okay? More activity in people with tinnitus compared to people without tinnitus. All right, and so let's take a look at what we've got in total. We've got hyperactivity in that pathway, in the pathway mediating the acoustic startle, in the pathway mediating the later waves of the auditory brainstem response. There's a lot of red. And I should say, actually, what's nice is that there is some corroboration for seeing elevated activity in these particular cell types, okay, or in the pathways that follow from them, that comes from the animal data. So remember, we're looking at responses to sound. I wish we were looking at spontaneous activity. We're not. Animal people can look at spontaneous activity, and when they've looked in the ventral cochlear nucleus, I think this may be like the only look, but it's sort of been neglected. But what you find is that there's cell types that correspond, there's neuron types that correspond to the spherical bushy cells and the globular bushy cells, and the particular type of multipolar cell that's involved in mediating contralateral suppression of DPs. And in all of these cases, there's some amount of increased spontaneous activity relative to control, which is the white bar here, that is present. Okay, so we're not necessarily, this is a way of saying that even though we're looking at sound-evoked responses, elevated gain in relation to sound, that may carry over to spontaneous activity. Okay, so we got, as I said, there's a lot of red. Now, we've looked mostly at the ventral cochlear nucleus, and as I said, as far as the cochlear nucleus goes, the animal people have mostly concentrated on the dorsal cochlear nucleus. Supposing that these two things, what was seen in animals holds in humans and what holds, and that's actually the most important piece, I'm going to add the hyperactivity from the animal work in here, and also the fact that we've seen these elevations in fMRI in the inferior colliculus. Okay, so we've got a lot of activity going on in relation, a lot of extra activity going on in relation to tinnitus. The gain is turned up in a lot of different cell types. Some of that may arise from reductions in activity coming in from the periphery, perhaps from hidden hearing loss. But, as I said, we've got massive descending projections in the auditory system. And I mentioned Brett Schofield. He's somebody who's actually worked out point-by-point connections in one particular neuromodulatory system, the cholinergic system, and shown that you start in layer 5 of the auditory cortex. There's descending projections that go to the pontomesencephalic tegmentum, all right, a major source of cholinergic inputs to other auditory structures, including, basically everything we've been talking about, cochlear nucleus on up. It's also the layer 5 also controls the medial efferents of the superior olive that are involved in the contralateral suppression, and those medial efferents are a source of cholinergic input to both cochlear nucleus and to the cochleas. What's kind of interesting is this caudal pontine reticular nucleus is actually the recipient of whatever cochlear nucleus neurons are mediating the acoustic startle. So basically, to the extent that we know it, every cell type I was talking about and pointing out in the cochlear nucleus gets some form of cholinergic input. And perhaps rather than the elevations in activity arising directly from a loss of activity in the periphery, there may be a large top-down neuromodulatory influence. And it doesn't necessarily have to be from the cholinergic system. It could be serotonergic, those and other and basically other neuromodulatory systems. It's just that the cholinergic system that I showed you, its projections to the lower auditory pathway have been particularly well worked out. So, when I look at this picture, all right, and I'm looking at all of these different types of neurons that are showing a turned up gain. As I said, going in and trying to figure out how to turn down the gain in the fusiform cells or the multipolar cells individually, right, where the gain might get turned down well for this cell type, but the same drug that you give doesn't necessarily turn down the gain for this cell type. That may not be the way to approach this. A better way is perhaps to look at some of the drugs that are going to affect the neuromodulators, basically the neuromodulatory systems that are going to affect, say, this top-down influence. Yeah, all right. So let me move on to basically pointing ahead. So I've tried to show you some about what we think is going on in the auditory brainstem of people with tinnitus. And as I said, tomorrow, what I'd like to do is move up to cortical levels and using imaging and discuss what may be going on in these higher centers, which are as important, if not more important, than what I just described to you, and in fact could be basically participating in the top-down neuromodulation that I was just hypothesizing. And thank you very much for listening.